and gentlemen, thank you very much for coming today, and it's very good to see so many of you here. I'm Dr. Anna Duggan, I'm Chair of Brain. I'd like to just ask Malcolm Skingle to give this 16th Brain Annual Lecture. 16th? Using innovation to promote the 3 R's.
What I'm going to talk about is changing trends in pharma and the regulatory framework. Some of you know this, some of you won't. Innovative technologies addressing 3 R's needs, and I hate the same old, so I'm going to share with you some collaborations that we have with people where they may work, they may not, but I think they'll be interesting. NC3Rs, I was on their board for 3 years. I chaired their studentship panel, and I was on their Crack-It, Dragon's Den type thing on a couple of occasions. So I was going to talk a bit about the work that Pharma have done with them, and then a bit on human target validation, because I think increasingly we are going towards in silico and cell line stuff. We're always going to have to, I think it's always going to be a place in medicines development for animal research, but it's certainly declining, thankfully. So changing trends in pharma. So there's an increasing focus on biopharmaceuticals. All pharmaceutical companies have got biopharm in their portfolio now. We had a target of 20% of our pipeline by 2015 being biopharmaceuticals. That's already up at 24%. So we're ahead of the game on that, which has implications on the sorts of studies we do. Repurposing initiatives. If a compound has been all the way through animal testing and into early stage clinical development, then why would you not look to use it in whichever disease indication you could? And as we learn more about the genetic profiles of certain diseases and implications for certain signalling systems and receptor systems, we are trying old compounds for new approaches. Smaller data-rich clinical trials, and that's not only clinical trials, it's also animal usage. We try and get as much quality information as we can for the minimum level of stress, either to the human or indeed to the animal. Increased focus on rare diseases, because actually, when you actually unpick perhaps a monogenetic disease that not very many people have, it will often lead to information which then gives you more confidence with some of the more common diseases. And we've got a very big rare disease program. The desire to treat disease of the developing world, increasingly, I think pharma are doing their bit. It hasn't always been so, but I certainly think we are now. We've got a 126-man unit in Tres Cantos, just outside of Madrid. All they do is disease of the developing world. They also are an open innovation campus, so if you have a good idea on treating a disease of the developing world, then you can apply for funding to go there. You can get access to all of our chemistry, our Category 3 containment laboratories, and they work on TB, malaria and kinetoplastic disease. Increased externalisation. I, for the first half of my career, was a pharmacologist, first 20 years. The last 20 years I've been in a business development type role, and I work with universities around the world on behalf of our scientists, brokering research collaborations. Now, I've always done this because I know that adds value to my organisation. Well, in the last few years it's got really sexy, everybody's doing it. And the CRO, contract research organisation activity, the university activity, and the research institute and interaction with biotech is on the increase. So I've worked for Glaxo all of my working life. At the turn of the century, Glaxo Wellcome, one of the heritage companies, and SmithKline Beecham merged to form GlaxoSmithKline. And this is the animal usage in GSK facilities in the orange there over the last 13, 14 years. Set at 100% there, you can see that initially it crept up, and then since 2007 it's thankfully been going down. The grey blob sitting on top of the orange there is our CRO work, so increasingly more of the work is outsourced to contract research organisations. Now this is our graph, our slide going the other way. This is human biological samples, HBS stands for. I'm doctor of the slide or anything, I just took it from one of our reports. And this is showing that the spend on human tissue is actually increasing. This does not include blood taken from our own unit, so I give blood regularly for experiments. The Stevenage site where I work in Hertfordshire, they take 260 litres a year, which our scientists actually work on, and they do 40 over in the US. It doesn't include the fee-for-service type work. It doesn't include the collaborative research, contractual work we do. This is just the tissue that we actually buy in. And it doesn't involve clinical studies where we actually collect the tissue ourselves. As you know, there's a regulatory requirement to test potential new medicines in 2 species before they can be tested in humans. We are always going to need this, in my opinion. I don't think it's ever going to be fully in silico, not in my lifetime at least. And we need that to assess, limit and manage the risk to human volunteers and patients. So I'm cutting to the crux of it early on before I get onto the technology. I mean, we are massively regulated in this space. Anybody who has ever worked in this space will tell you, you know, you need a site license where the animals are held and where experiments are taken on. The Home Office inspector can turn up at any time and just demand entry, and he will check out things like the number of air changes in your room and your labelling and the number of animals per cage and anything he wants actually. And if he finds anything untoward, he's at liberty to actually close that site. So obviously, all for the right reasons, we take that very seriously. The project licence is where you will agree with the Home Office inspector the species, the route of administration, the specific procedure, the duration of any experiment, line by line, wholly with in mind the 3 R's principles. I once wrote the neuropharmacology Home Office licence. I swear it's the hardest thing I've ever done, the hardest thing I've ever written. Made a PhD look like a piece of cake. And then you need an individual licence. Training, graded approach to licensing. You're not let loose until you have been trained. If you change project areas, you need to change your licence. Everything you do is licensed. So there are three levels. And the whole licence procedure sets out the benefits of doing the work, outlines and illustrates how 3 R's will be implemented as a part of that process. So these are the Home Office returns for 2013. More than 4 million scientific procedures using animals in 2013. Rodents accumulated for more than 80% of those. Rodents and fish, you know, danio, zebrafish, are being used increasingly for lots of quite complicated, even signalling pathway experimentation. So together they account for 93%. Dogs, cats and non-human primates, less than 0.4. We've actually got an office, and have had for some time. In fact, until I researched for this talk, I didn't realise we've got this many people in it, to be honest. We've actually got 20 people working around the globe, operating under several regulatory regimes, who are upholding global core principles and also looking after the compliance with the local regs. Things like the governance of the project licences, but we've got a very clear mandate which is set out in our corporate responsibility report every year, and they make sure that we stay honest to those. They also monitor the contract research organisations. We don't think just because we've got a contract in place, we've lobbed it over the wall and we don't have to worry about it. They can't even start any work until one of our vets has been in and actually assessed the animal husbandry, and it also goes for universities. So every contract that my team put in place with any university in the world, one of our vets has to visit there and sign off on it before we can embark on those studies. And they've done 600 assessments over the last 5 years, which surprised me. I didn't realise it'd be that many, but when I come to think about it, it's probably about right. And the studies must satisfy our core principles of animal care and welfare. And they record those numbers. It's just not to, as you saw, we count the numbers, it's the grey area on the orange bars there. We actually record those and we report on those each year. We also match those up to the programmes of research in the agreements that my team put in place. Excuse me. So we have a corporate commitment to animal welfare. We do publish it in our corporate commitment report, as I said, every year. I think we were one of the first companies, our chief exec banned the use of great apes. Not I think we use them much anyway, but Non-human primates only used when no other species is appropriate, and there are some occasions where we have to use non-human primates. And we are wholly committed to sharing animal-based data. And I would suggest to you that GSK are one of the more open companies when it comes to sharing all manner of data. Would be my experience, having set up many consortia agreements across the industry in the last decade at least. And actually, you know, when I think 10, 15 years ago, it would have been laughable to think that we'd be working with Pfizer and AstraZeneca and Novartis on some of the programs we're currently working on, and that's pretty much commonplace. In fact, here's quite a few public-private partnerships. These between anything between 2 and 25 partners on these consortia. Some are IMI type stuff, Innovative Medicine Initiative, which is €1 billion from Europe and $1 billion in kind from Pharma. And they're working on things like translational safety biomarkers, for example. Where there is value in actually pooling our data, and certainly pooling our data on toxicology in, say, liver or kidney. There are other consortia for that. And we have similar things in the United States. Now, pre-competitive collaboration obviously avoids duplication of data generation. You get greater stats. In fact, we're finding this even with our human genetic studies. We used to be so arrogant as an organisation, we used to think that we could do all the science that we needed to be profitable and to do really good science as we do. And actually, we need to pool our masses of data with other people's masses of data, particularly when you're looking for very rare incidences of genetic differences. And from an animal perspective, of course, this would reduce attrition, and it also leads to smaller, better informed clinical trials and ultimately improves on patient safety. So onto the sort of technology bit. I could have chosen any one of several dozen things, but I've decided to choose, knowing that there's a few Nottingham connections. Nottingham get a mention, and the work we do at NPL, so I'm going to talk a bit about that. So the two most important things you wanted to know about any drug program is, does the compound actually get to the target, and where the hell does the rest of the drug go that doesn't actually get to the target, which is often a significant amount, and does it accumulate anywhere? And depending on the type of molecule and the particular disease you're treating, it could be a pessary, it could be a suppository, it could be under the tongue, it could be inhaled, it could be oral. You want to know ultimately that it gets to the target in sufficient amount to actually do its stuff, not stay around for too long, to possibly lead to accumulation and a toxicity, but it's long-lived enough to actually have its beneficial effect. So for every exploratory medicine, you want to know, does it get to the target cell? Does it get to the target site within the cell? How much gets there? And then you want to know which tissues other tissues does it get to, and does it accumulate there, and is that likely to cause any bad news when we get later on down into the clinic? So where does the drug go? The vast majority of the drug does not hit the receptor that you want. It's going to be elsewhere. And I just put this slide up just to say that you can do all the modelling that you like. At the end of the day, you're going to have to refine those models, and it's going to be based on biological data. So you want a compound that's high potency, with high affinity, that gets to the receptor site, that does its stuff, doesn't stay around forever, and then depending on that, you can work out an SAR and a time course relationship. One of the techniques that we are increasingly using is mass spec imaging, because what you can do with mass spec imaging and some very, very clever algorithms is you can actually see where your drug gets to, and this is going to be the Nottingham connection in a moment. So there's a couple of techniques there. MALDI, Matrix Assisted Laser Desorption Ionisation. I actually wrote it on the bottom in case I forgot what it stood for. And secondary ion mass spec, SIMS. And this is where you would have a tissue sample, and you would actually use a beam to actually slough off and get a mass spectrum, which you then can record, and then you raster, which you move very slightly one way or the other, up or down or from side to side. And you would go along in a grid-like fashion, and you'd record thousands of mass spec like that, from each position, and then you deconvolute that, and you reconstitute using algorithms the actual slices in real time and where your drug was in those slices, and possibly even the metabolites and the endogenous substances in that space. Because we now have libraries which have got most of these things in, and you're getting the masses down to such finite resolution. So this would be a typical MALDI mass spec imaging workflow. So in the first instance, you would take a slice, you would spike that slice with your compound in decreasing concentrations. You would then see what the detection limit was using this technique. And if you could detect it in a sensible time frame, you would then dose your animal with that, the dose range I should say, not time frame. You would then dose your animal with that, and you would take those slices, and then you would use this technique to determine whether or not the molecule actually got to where it needed to be. And then you can generate, I won't bother going through this in the interest of time, but you can go through this process, and you can do some quite sophisticated things just using one or two rodents. So this is the drug distribution in the rat lung using MALDI MS, and you can see that the figure on the bottom left there is a 200 micron resolution, and the lighter stuff here is the drug. And you can't actually see from that at 200 micron resolution whether or not the drug's actually in the cells, in the tissues, or whether it's in the airways. When you go to the higher resolution of 30 micron, you can see very clearly that it hasn't actually left the airways. And so it's giving you more information. As these techniques become more and more refined, which is exactly what we're working on, we will be able to get more and more information from each of our slices. So safety assessment can use it. So here's a good example where you have to do a 14-day tox study, 0.5 milligram per kilogram per day of a nebulized solution. Day 14, 1 hour after the final dose, the animal is sacrificed, slices are taken. The left-hand side slide there is for MALDI, the mass spec technique. The right-hand side is stained with eosin, and it's a histological stain. And then you compare the two. So the MALDI information, just to cut to the chase, shows us that the white bits here is the compound accumulation, which again is reinforcing the fact that it's predominantly in the airways. But the bad news is that at quite a low dose, this staining here, the darker purple in exactly the same areas, means that the compound's actually causing inflammation. So a dose that would not be detected by looking at the phenotype of the animal, you can actually detect using this, so this compound would be shelved straight away. So the animal is at far reduced exposure to potential risky molecule. You can also use it for efficacy studies. So this is another respiratory study. The clinical candidate there, you can see, is restricted primarily to the main airways of the lung, whereas the backup molecule is spread more evenly throughout the lung tissue. You can even use it for domain antibodies. So we acquired a company called Demantis back in 2006 for 230 million to get to this next generation antibody technology. So these DABs, as they're called, are 1/10 the size of a full antibody. You can actually dose these up, and you can show these using MALDI MS. And on this slide here, this is in a flu model. Okay, and you can see that 1 hour after dosing a control animal on the left and an infected animal on the right, they pretty much look the same. You can't really discern a difference. When you go to 24 hours later after that dose, you can see the domain antibody is cleared from the control lung, but it's still in the infected lung, which gives us the confidence to know that it's where it should be. It's probably doing what it's supposed to do, and it gives us the confidence to go on to the next stage and progress with that clinical program. So we can use MALDI imaging mass spec for all manner of tissues, depending on the disease we're looking at, and indeed we have. And we've done it. We've done MALDI analysis using a single animal in lots of tissues, again reducing the number of animals that we would use. So our current position is MALDI is used routinely to look at the spatial distribution of molecules. If there's anything I say that I don't make clear, or I go into acronym mode, just put your hand up and I'll stop and explain. And then you can use it for small molecules, you can use it for DABs, you can use it for dose ranging studies to find the optimal dosing level. You can use it to look at differences between methods of dosing, which is sometimes important to us, and importantly you can correlate efficacy and toxicity. What you can't yet do is see specifically which cells the compound are getting into. You can get to the rough area, but we want to get better than that. We want to get to actually which organelle does our drug get to, and there are some of these organelles within the cell. Whoops, whoa, 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 whoa. Fat fingers. There are some of these cells that, some of these organelles actually that you don't want to get to, because they're going to be indications that you are likely to have a problem, and there's likely to be a toxicity, and there's others where you definitely do want to get to. And they're of the sorts of sizes, you know, mitochondria down at a micron there, and you've got lysosomes at 250 nanometers. We want to get to that level of resolution. Now, there's not one single organisation who have got the instrument to do this. You've got some brilliant physicists, you've got some great metrologists, you've got some great biological scientists and people who know how to handle tissue, but we haven't actually got them all together. And that's the fun part of my job in helping bring people together from premier institutions to actually drive stuff forward. I really get off on it. So we want higher spatial resolution, we want higher mass resolution, we want an ability to maintain tissue and cellular morphology. A lot of stuff in the old days you'd have to use fixers. You can't use the fixers for MALDI because it displaces the drug. So you have to think about how you do that. And then you also need a high optical resolution system, probably electron microscopy, to actually bring the things together so you can do overlay maps. And we think we've pulled together the group for that. So we've got a professor in Gilmore who gave me some of these slides from National Physical Laboratory. One of our guys, Andy West, who's a mass spec expert but really knows how to handle biological tissues. There's two German companies, unfortunately. It'd been better if they were British, but they were the best. So Iron Toff, who are mass spec people, and then Thermo Scientific, who've got the Orbitrap mass analyzer, which is the best. And we're trying to take the best of each of the groups, and then we've got the University of Nottingham, First Link, where we've got three of their professors advising, and a chemist from University of Chicago, who's also advising on the project. And you need the experts. There isn't any single institution, GSK included, that's got all the scientists it requires under its roof to actually drive its science forward. So we had a press release back in December. Well, this is kicked off now. It got a £4.5 million Strategic Capability Programme grant from BIS, from the National Measurement Programme. And we're on the way to doing this now. And I haven't got time to go through these, but apparently this is pretty impressive. We can show our drug in villi. You can actually use the software to actually rebuild from all of those mass specs that you've got. You can actually rebuild, and you can actually show specifically where the drug is actually getting to. So it's a 3-year program. I think at the end of 3 years we'll have some good stuff to talk about. We don't own any IP. We just want access to it. They don't know how to do the biological samples. They don't really know the biological questions to even ask. We're helping out with that, but we will take the first 3 or 4 big bits of kit that come off that production line, and then hopefully our competitor companies will also take up the technology as well. The second example I wanted to show was, it's always been the holy grail to get human hepatocytes to do drug metabolism studies. You can never get enough human liver. People want to keep their livers, apparently. And we are now getting to the stage with our knowledge of pluripotent stem cells and generation in serum-free media. We're now getting to the stage where we can develop cell lines that are of use to us. And I always think it's when people who are experts in their own field get together and have an open conversation with experts from other fields. The chemistry guy here, Mark Bradley, I've done shed loads with over the years, really good guy. Used to work down in Southampton, then he got poached by Edinburgh. He's been up there for quite a few years now. But he's into combinatorial chemistry. And I first interacted with him probably 15, 20 years ago when we thought combinatorial chemistry was going to be the saviour of the industry, and nothing could be further from the truth. But he thinks in combi-chem terms, and he thinks in terms of combi-chem materials. And he's now applied this logic in polymer combi-chem to stem cells. And he's working with David Hay from the MRC Regenerative Centre. David's been working on hepatocyte cells for some years. Met with Mark. The gold standard at the moment is Matrigel. You grow your cells because you want your cells to stick on Matrigel for long enough to survive and be effective for you to squirt a drug on it to see if the drug's getting turned over, whether or not it's getting metabolised. And it's always been that enzymes like CYP3A, which is an important one, which metabolises it does metabolise half the molecules that are out there on the market as medicines at the moment. You want to know if CYP3A is induced by your drug, and also whether CYP3A chews up your drug. And on Matrigel, there is a phenotypic instability, i.e. the cells change with time and they become less useful for you. So you had to get them just about right. So what Mark did, he actually looked at several polymer microarrays, converted things to 384 well template, actually used printing to actually put this stuff down. And to cut a long story short, they then went to the next stage of putting the cells on top of the arrays, incubating, washing the slide to see what sloughs off, because obviously you want the stuff to stick, then fixing and imaging to see which ones were viable. And here's half a dozen code names for different polymers, but this one on the end here, 134, or U34 was it? This one looked pretty good, using the markers, and importantly it had 6 times more of one of the CYP1A enzymes than any of its competitors. And then looking over a range of hepatocyte metabolic activity, they found that they could actually, with serum-free media, actually get the cells to grow and be comfortable in the presence of the new polymer that Mark had produced. And this is just to say that when you compare it to primary hepatocytes, where you've actually prepared the cells, that the stem cell derived hepatocytes do at least as well, with all of the, well not all, but a whole list of 20 compounds there which are known to interact with liver cells in DMPK ADME situations. So this is work in progress. The earlier work that I showed you is work in progress, but these are one of many programs that we have where we are trying to look at alternatives to using animals in research. Oh, that's just an experiment I forgot I put in. So, yeah, so they flourish. The pluripotent stem cell derived hepatocyte cells actually stick to the novel polymer. The CYP enzyme levels are maintained over time, and this now opens up the possibility of doing even chronic studies. So as before, the cells didn't live long enough to do chronic studies. They died before you could get to that point. And you can actually do mechanism of action type of experiments using these. So hopefully they'll be publishing on that quite soon. Now, we don't actually collaborate with these guys as yet, but this is the sort of thing that people like me keep a watching brief on, and sort of certainly scientists within GSK, so that as soon as one of these technologies gets to the fore, we would then internalise it and use it. We'd either license it in from a biotech or indeed collaborate with a university. I wanted to just talk about the pharmaceutical engagement with NC3Rs because I think they're 10 years old now. I came off their board last year. I did my 3-year term. The ABPI have worked with our trade association, Association of British Pharmaceutical Industries, have worked with NC3Rs since it started. And I think they're still funding a post there. I chair their studentship panel because I have a lot to do with students and academics. And I was also on their Crackit, which I'm going to talk about in a sec. I was also on their Crackit Dragon's Den thing, which was great fun. So they've been going for 10 years. They've been tasked by government to work with the science base. I think the best thing they do is they act as honest broker between lots of stakeholders, which is great. They get people talking, which is great. So certainly across pharma companies with biotech companies, academic companies who've got academics who have then got spin outs, academics who want to work with pharma, academics who want to take stuff all the way through on their own. I think they've got the networks to be able to help do that. They now get significant funding because they did well with their initial funding. Of their 20 staff, a dozen of them were scientists, and of course they work also with the agencies that the pharma companies would have to work with, which is good, so we all start talking a common language. So they're looking for new models, they're looking for new tools and approaches, they're looking to nurture tomorrow's leaders, influencing decision making in a fair and unbiased way. I like to think that they work with companies, but they're not in the pocket of companies. In fact, I was having this conversation with Bob just before we started. I think the independence is absolutely great, and of course raising awareness. I mean, their figures on their web hits and downloads is significant. So I'm a fan of NC3R, starting from very low knowledge of actually what they were all about. So the networks I've mentioned. They have a number of funding streams that people can tap into. They've got 14, I think it is, working groups. I've underlined a couple here because I'm going to say a bit more about each of these. But certainly we're involved with lots of these working groups, and certainly we were involved in the 4 that were underlined there. So the first one is the acute toxicity, which we all knew was a dreadful waste and a useless waste, frankly, of animals that, because of the regulations, we had to do. And really, it was GSK can't take the upfront credit for this. Sally Robinson, AstraZeneca, was the champion for this, and the other person to, I think, point out in this is Catherine Chapman, who's on a little video clip in a sec. But Catherine actually pulled the networks together, and you can't read those company names probably, but all the big companies are on that list. And they asked the question rightly, do we really need single-dose acute toxicity studies? What do they really tell us? So they got 18 European companies together with the CROs through NC3Rs, shared data over 70 compounds on the number of animals used, routes of administration, excuse me, how the studies informed internal decision making, and to cut to the chase, actually they didn't. And looked at the considerable variation in study design. And the main conclusions that came out of that was that the value of the acute tox study was limited. Certainly wasn't used in setting doses for animal studies. I mean, often, having worked in the pharmacology department, sometimes it would be the solubility of the compound, which was the limiting thing, which is obviously ridiculous. They certainly do not provide any information on the nature of the toxic effects, and they're not used to set the first dose in human. Because there are other studies, frankly, that give you more valuable information that you would use in that decision-making process. So the output was, certainly in 2004, there were 11 companies there who actually had 2 rodent species using 2 routes, i.e. 4 studies, which is what you had to do. And by 2007, that had just gone down to a couple of companies who actually were actually putting those into their submissions. And this down the other end here, because you can't read the writing, it's not my slide. The light blue, there is no studies as a default position, and certainly GSK were one of the first to drop the acute tox study. And in terms of animal use, that's a 70% reduction in animals. And it's not on the slide, but I think that's supposed to equate to something like 15,000 animals. So, and this was just NC3Rs helping pull together with a champion. You always need a champion for these things, and it's best actually that it comes from the industry, and it did, and then NC3Rs helped pull it together. What it meant for the regulatory change to ICHM3 was that the proportion of clinical trial applications using acute studies has gone down since 2007, from 86% to last year, which was 16%, and certainly ours, GSK's didn't have any in there, that's for sure. So most people get it because the information got out there quicker, and people are using it. Now that is a fairly blunt instrument. I would suggest to you that most of them aren't quite like that, and all the time we are refining to adopt the 3Rs principles in all that we do. So the next one is ARRIVE, Animal Research Reporting in Vivo Experiments. Hands up, who's heard of that? About half, okay. So basically the bottom line for this was there were some poor publications out there, you know, really. And there was a survey done quite some time ago now. Carol Kilkenny was the first author, looking at the quality of the experimental design and the stats analysis and the reporting of the studies from UK and US publicly funded research. I think it was 271 papers, something like that. It was quite a lot. Looking at the experimental design, most papers did not report randomisation, 88%, or blinding, 86%. You know, that's criminal. I mean, 30 to 40 years ago, I was doing that. It will bias your results, particularly if you're doing behavioural experiments, if you don't randomise and if you don't actually blind which compound has gone to which animal and then deconvolute the results once you've finished. Statistical analysis, only 70% of publications fully describe the stats. I mean, what the editors and the reviewers were doing, I don't know, but that's how it was. So, which meant that 30% didn't put good stats in. And then only 59% included the three important pieces of information like the hypothesis, number of animals and the characteristics of the animals. So obviously people needed to pull their socks up. Most of these, I hasten to add, in fact probably, yeah, the vast majority would have been academic papers because they received funding from the UK and US authorities. You know, we don't. So what happened was the ARRIVE guidelines were developed in typical NC3R fashion. There was a great deal of buy-in from all the key stakeholders. They worked with people in the field like the statisticians and then the charities and then the journal editors. And the outcome for that was that now the Nature papers in 2013 have to adopt the ARRIVE guidelines. It means that the funders like BBSRC and Wellcome Trust, MRC, wrote to all their grant holders and saying, hey, you need to look at these guidelines. It's a way of getting it. There's no argument against it. You know, no one wants to read poor science. I find it hard to believe that anybody would want to publish poor science, and you want to get the most bang for your buck. You want to get the most kosher information that you can from a limited number of experiments. So I would suggest to you that that has been a great success. Now Crackit's a bit different. Crackit's a bit wacky. Crackit is where the actual problems can be posed by Pharma. You know, if we say there's a real need, and we have, or biotech, or sometimes they bubble up from the academic, but it's obviously the whole focus is on reduction, refinement, replacement of animals in research. What the NC3Rs do in quite a professional way is they broker the relationships, and sometimes that will take several iterations even before anything is submitted for potential funding. Then it goes through the normal aggressive and bloody peer review that all poor academics have to go through whenever they attempt to get money. And then the thing is actually project managed by NC3Rs. It's not one of these lob the money over the wall and we'll have a report in 2 or 3 years' time. It's actively managed and it's milestone driven, pretty much like most of the contracts that I would put out now on behalf of GlaxoSmithKline. You know, actually if you don't get to go and you don't pass go, you don't get 200, you don't then move on to the next stage. We would actually close it. So there has to be seen to be some delivery and some progress. This is one example, Robin Williams, not that Robin Williams, from the Royal Holloway, who developed a slime mould model of emetic liability. We were interested in that because we don't want people to feel sick if they take our medicines, so we wanted some indication of that. But also Syngenta were interested, but they were interested from the other angle. They wanted it such that if a critical dose of herbicide was ingested, you actually vomited it up. So they wanted it for completely different reasons. But we both provided chemical libraries into the programme. We actually provided some funding, and this was one of the things that we were trying to use, because obviously we want to go as low down the sentient animal organism chain as we possibly can.
Crackit Solutions was developed to accelerate the uptake of new technologies and approaches with benefits for the 3Rs up into the marketplace. Alex Easton has found through Crackit Solutions that he's been able to raise the profile of his technology and also find new partners, including large pharmaceutical companies like GSK.
We've developed a piece of apparatus that allows us to reduce animal numbers in a really common test of memory. But it's most widely used as a test in industry, and we as academics had no idea how to get into industry and find out how they use it and so on. So we wanted to get into Solutions so they could tie us up with a partner. And the great thing about Crackit is that the NC3R's team helped me put together a pitch to make sure that industry would be interested in what I had to sell. They used their extensive contacts to help me link to people. But once they'd given me the link, they stepped back and let me talk to them directly, so I kept complete ownership of the idea. So we want to build a partnership with industry, but sometimes it's difficult to get on board if you've got long-term investment needed in getting things going. The beauty of the Crackit solution was that there was some money we could apply for that has allowed us to kickstart that project and actually work. Already we've got an apparatus built, we're in contact with a partner every month, and we're really starting to see how our apparatus can fit in that industry setting. Working with GSK over the last few months, we've been able to better understand what their needs are and therefore be better able to put our apparatus into industry and have the savings in animal numbers that we're looking for.
So that's from the horse's mouth. He didn't know how to get into a company like GSK. They helped broker it. So it's been a good program for us, and I would suggest it's been quite a good program for the academic base. This is another one. I haven't got a video clip here, but this was one that I was actually on the Dragon's Den for. And this is an academic, Douglas Armstrong, who is at Edinburgh University, who's spun out a company, Actual Analytics. And this is just to do with locomotor activity, but it's then getting to the next stage of refinement. So this is with cameras and transponders, and it's iterative. So it's about social interaction when there's more animals than one in the cage. It's about stereotypy, stereotype behaviours, temperature regulations, looking at circadian rhythms, and this is a program which is going to be a commercial success. Most of the programs are using SMEs, small medium enterprises, who've got a vested interest in actually delivering a product at the end of it, which hopefully then we'll all tap into. This is actually being driven by a chap called Redfern at AstraZeneca. So it's launched in 2011, there have been 16 challenges, high SME involvement as I said, the money's reasonable, you can do stuff with the amount of money they've got. It's captured the imagination of the large industry sponsors and also the funders, and we're actually involved with one of this year's challenges, that top one, to generate physiologically relevant human stem cells to identify neurotoxicity. Now, you don't want to do that in vivo, you want to do that sort of stuff in vitro, but you need it to be reproducible and you need the assays to be viable. So this is just to say that GSK sponsor the 3R prizes, and I just wanted to put this one up just to remind me to say that their reach is even out with the UK now. The tall guy on the right hand side there came over from the US to collect his award. The lady standing next to me there, she was working in the Netherlands and in Cambridge, Gurdon, and she's also working on hepatocytes. The last little bit, I just want to talk about this is a personal view, but I think that increasingly the people who will be valuable to organisations are the people who have done science, are bench scientists, but actually are people who can actually take disparate pools of information and knowledge and create new knowledge from those and see things that other people can't. Actually develop the ontologies from anything from genetics through to filling chemical space through to screening results and screening data from high throughput screens. We embarked on another different collaboration earlier this year. We call it the Centre for Therapeutic Target Validation. It's between the Sanger Institute, who did the human genome, or part of the human genome, EBI, Emble EBI, who are right next door, and a field in Cambridgeshire, for those of you who don't know, in a place called Hinxton. And it's getting to the stage now where the cost per genome is coming down and down and down from when the first genome was produced, and we will get to the $1,000 genome in the next few years, I would have thought. It's now getting to the stage where we've got so much data, there's so much information being published in biological sciences, you can't actually keep up with it. You need smart algorithms to help you with that data. And I think that genetics, probably a decade ago, was overpromised, and we thought it was going to be the saviour of everything, a little bit like CombiChem 10 years before that. And of course it hasn't been, and I think that has made certainly some pharma companies a little bit reticent to shed loads of money into that as a development area. But I think that it's now changing. I think the sequencing costs have come down, the algorithms are so much better, our understanding of biological mechanisms, we now know far better what we don't know. And so we've set this centre up. It's going to be 50 people. We're going to have 2 of our teams working actually in Hinxton, actually embedded within the group. It's all about open access to tap into big data, which everybody's talking about. And we will, it's multi-million funding from our perspective, but what it will do in effect, it will help us de-risk our research by combining data and expertise. And obviously the Sanger are very much into the high throughput sequencing and phenotyping. The science team is made up of the top there are EBI people, then GSK people, and then Sanger people on the bottom. They're all world leading in their own right. They will all bring a different perspective to the problems that we're trying to address. And we will take all of the publicly available data, of which there is absolute shed loads. The people at EBI are very good at curating and mining that information, and we will have a core database that we will mine collectively. The Sanger are great at generating some of this data, and then we've got our teams who are posing the question, say, well, can we get to this just through the in silico information that's currently available? And I think this is really exciting, because we will get this up and running. Already I've had a couple of other pharmaceutical companies write to me and say, hey, can we come into this? We are in some consortia already, things like the Structured Genomics Consortium, which is, again, 10 years old. We started that off with the Wellcome Trust a decade ago. There's now 8 companies in that. People are coming round to realising that if you put more information out, you get more back. And so this slide was given to me by you and Bernie, thanks very much, from EBI. And I didn't realise it did this, but if you look at the blue along the bottom there, the academic open stuff, when you click the button it goes to more blue. We're trying to make more of the stuff available to more people, because actually if you do put it out there, then other people start to think about your problem. And we do it in lots of other areas. You know, why don't we do it with target validation? So we're going to mine big open data, we're going to really tap into human genetics in a big way, and we're going to do lots of human cellular experiments, which are going to then, in an iterative fashion, feed back into that data set when our scientists pose the questions. This is my last slide. In silico methods and modelling will become increasingly important, I believe, in drug discovery, when they are now. High quality information from data-rich experimental animal and human studies will shape future regulatory packages. I think in the past we made a rod for our own back. We kept making our regulatory packages bigger and better, and actually that was then the new standard, and then they just get another stack of paper of data. I think that we will get smarter. We've got some pretty smart people. We've got some smart people we're collaborating with. We'll get smarter about how we pull those packages together. The use of human tissue and cell lines will increase as animal usage decreases, and certainly the industry will continue to innovate and partner to address the 3Rs challenges as much as we can. I hope I've convinced you of that tonight. That's it. Thanks.